Welcome to my YouTube channel. I'm by the name of Josephine Adukun. I'm a student at NS Ruhengeri studying the biomedical laboratory science. And now we are going to be together discussing about the antigen antibody interaction. Talk about the antigen antibody interaction. In this session, we have the 5 questions we are going to focus on. We are going to start with the first question, which asking what is the principle behind the agglutination. First of all, we are going to know what is the agglutination first. Agglutination is the interaction between the antibody and the particulated antigen resulting in the visible cramping. The interaction of the particulated antigen with the antibody led to the agglutination reaction. Agglutination reactions are the word used in the laboratory diagnostic for identifying the blood type, detection of the bacterial infection, or identifying the specific antibody or antigen in the sample. There are the different types of the agglutination based on the particulated particle which is involved. We have seen that the agglutination, there is interaction between the antigen and the particulated antigen with the antibody. Now, based on the type of the antigen which is involved in the interaction, it can be classified in the two types. There is direct agglutination and indirect agglutination. When you're saying the direct agglutination, it occurs naturally when the antigen are present on the surface of the cell. For example, during the blood typing, as you know that the antigen, it is mostly found on the cell, on the red cell. When the antibody in the antiserum, it can bind with that antigen, it can form the cramping. That is what you call the direct agglutination. Then indirect agglutination, to test the patient's serum for the presence of the antibody against the soluble antigen. In this, the agglutination, we are looking for the antibody which is produced in the body, which is going to fight against the soluble antigen. When you are saying the soluble antigen, it is here the soluble antigen are the antigen that are not bound to the cell surface, but are instead found in the soluble form in the body fluid. For example, found in the blood, in the lymph, or the tissue fluid. This antigen can be protein, polysaccharide, or the other molecule that are capable of the triggering the immune response when they detected by the immune system. That the soluble antigen is that antigen which is not found on the surface of the cell, but is able to trigger the immune system to produce the immune response when it is detected. The serum is mixed with the latex sphere with the soluble antigen attached. That means that on the latex there is the soluble antigen which is coated with. The antibody from the serum of the patient will then cause the visible agglutination of the latex sphere with the soluble antigen. Alternatively, the antibody may be attached to the latex sphere. For about the latex, sometimes it is coated with the antigen when we are going to looking for the antibody in the patients, or alternatively, it can be coated with the antibody when we are going to looking the soluble antigen which is present in the serum of the patients. That is the difference between the direct and indirect agglutination. The principle behind that agglutination reaction is based on the antigen-antibody interaction. In this reaction, the specific antibody binds to the particulated antigen in a way that causing the visible cramping. That means that it is not all the way the antibody-antigen bind. In the agglutination, the antibody, it can bind to the antigen in the way causing the visible cramping or the aggregation of this particle to making the cramp. This visible cramping formed is called agglutination. Here there is the image which can showing the how agglutination reaction occur. Here that is we have the bacterial cells and there is the antibody. When that antibody binding to the epitope or the antigen determinant and cross-link that cell, it can forming what you call agglutination. Here there is the clear example which can clarify it very well. Here there is the antibody. When that antibody binding to the antigen finding on the cell, and when that antibody cross-link the antigen and can forming the clamp, that's what you call agglutination. That antibody which involve in formation of the clamping is what you call agglutinin. Here there is the other example, which can showing how the antibody antigen interact and form the cross link in between the two antigen in order to forming the clamp. That is the first question, the saying about the principle behind the agglutination, and we see that the principle behind is antigen antibody interact in order to detect whether the patient has the disease or there is no disease. For the question number 2, it is asking, in the agglutination we use the latex bed, and we are going to see how the latex work in order to diagnose whether the patient has disease or not. The latex agglutination, it can work in a diagnosis. It can work in a diagnosis by using the latex bed coated with the specific antigen or the antibody to detect the presence of the corresponding antibody or the antigen in the patient's sample. That means that in this the latex agglutination, the latex bed, it is either coated with the antigen or the antibody which is corresponding to the antigen or antibody we are going to looking for in the patient's sample. During, for example, when we are going to looking at the antibody or antigen in the blood, in the urine, or in the cerebral spinal fluids. For this, the latex agglutination, in order to be performed, it can take the step by step, and we are going to discuss one by one. The first one, it is coating the latex bed. Here, that latex bed, it needs to be coated with either antibody or the antigen based on what we are going to looking for in the patient serum. After coating it with the molecule, it mixed with the patient's sample. Here, we took the patient's sample and pour on the latex bed which is coated with the either antigen or the antibody. If the sample contains the corresponding antigen or the antibody, the binding it occur. But when there is no corresponding the antigen or antibody which is found in the patient's sample, there is no binding taking place. The third step is the observation of the cramping. After antigen and the antibody bind in the way which is causing the cramping, when the target antibody or the antigen in the sample bind to the latex coated particle, the visible cramping or the agglutination, it will occur. This indicating that the patient has the target antigen or the antibody. That is the third step where we see the cramping based on the antigen antibody interaction. For the last stage is cross-linking. That antibody antigen interaction, in order to form the cramping, it needed to cross-link the antigen. When the antibody bind to the antigen on the multiple particle, they form the cross-link between the particle in order to forming the aggregate or the clamp. Each antibody has at least the two antigen binding sites, so it can bind to antigen on two different particles. That means that when that antibody have two antigen binding sites, it's able to bind to the antigen on the two different particles simultaneously. Here there is the image which can showing how the latex agglutination it can occur. Here we have seen in this image the latex bed, it is coated with the antigen, and when we put the patient sample which contain the suspected antibody, it can interact with the antigen which found on the latex bed. Then when the cross-linking occur, it can forming the agglutination. When the particles are coated with the antigen, agglutination indicating the presence of the antibody, either it's like the IgM. In this image also, it can show when the latex bed is coated with the antibody, what we are going to looking for in the patient sample. Here in the image, it can show that we are going to looking the antigen which is present in the patient sample, while the latex bed, it is coated with the antibody. When you see the agglutination, it means that there is a positive result because there is a presence of the antibody in the patient sample. Then let's talk about the third question, which saying that what is the principle of the precipitation reaction? First of all, when we need to know the principle, we have to know what is the precipitation reaction. When we say the precipitation reaction are the reaction in which the antibody and soluble antigen interact in the aqueous solution. That means that here that soluble antigen and the antibody, it needs to interact in aqueous solution in order to form the latex that develop into the visible clamping. That latex which is formed is one which is coming into the visible precipitate. The formation of the antigen antibody latex, it depends the nature of the antibody and antigen, either the variance the antibody or the antigen has. Firstly, antibody must be bivalent. It have the two antigen binding sites. And the second, antigen must be either bivalent or the polyvalent. It must be able to bind with the antibody more than one antibody. That is the property which can help the precipitation reaction to taking place. The precipitation occurs when the antibody mixed with the soluble antigen in equal proportion. In the previous question, you have seen that the antigen-antibody interaction, but it is not necessary to be in equal proportion. But here in precipitation reaction, it is mandatory to be in the equal proportion in order to detect the quantity of the antigen which is present in the patient. When the antibody mixed with the soluble antigen in equal proportion result in formation of the insoluble immune complex, which is called the precipitate, and that precipitate formed, it is visible. Then the principle of the precipitation reaction in immunology is the based on the specific interaction between the soluble antigen and the antibody, but in the solution. It is not like in agglutination, but here it's taking place in the solution and leading to the formation of the insoluble complex that precipitate out of the solution. That insoluble complex formed, it precipitate out the solution. The reaction, it occurs when the antibody bind to the antigen, as I said, in the optimal proportion. Here there is the precipitation reaction in the fluid yield a precipitating curve. Here there is the pro-zone, zone of the equivalence, and the post zone. Let's discuss the explanation about this graph. The excess of the either antibody or the antigen interfere with the minimal precipitation. That means that when there is the high antibody or when there is the high antigen, it can interfere the minimal precipitation, which taking place in the zone of the equivalent, where the antibody and the antigen is the optimum. The interaction of the soluble antigen with the antibody, either IgM or IgG, it can lead to the precipitation reaction. That precipitation reaction, it depends on the formation of the latex, and it can occur best when the antigen antibody, it is in optimal or equally. That is what we call the precipitation reaction. That means that here, in order the precipitation reaction take place, the first, it can take place in the solution, and it can take place when there is in the equivalent zone, when there is the high antigen or the higher antibody. That means that the precipitation reaction, it cannot take take place. Agglutination reaction and precipitation reaction are both based on the antigen-antibody interaction. They are principally based on the antigen-antibody interaction, but they differ in the nature of the antigen involved. They differ on the visible outcome and their diagnostic application. Here there is the image which can showing how the agglutination reaction look and how the precipitation reaction look. Here in the agglutination, there is no need to be in the solution, but here in precipitation reaction, it needs to be in the solution, and there is this precipitation reaction, it needed to be in equivalent zone. Let me see the difference between the agglutination and the precipitation. The first of all, when you based on the antigen, in the agglutination, it involves particulated antigen, that antigen which is insoluble. In the precipitation reaction, the soluble antigen is one which is involved. Based on the antigen-antibody complex, here in agglutination it can forming the clump or the aggregate, but in the precipitation reaction it can form the visible precipitate in the solution or in the gel. Based on the outcome, the clumping, agglutination reaction, it can form the clumping particles, but in the precipitation reaction, the formation of the insoluble lattice, which is appear as the precipitate. According to the optimal condition, this agglutination reaction can occur over a wide range of the antigen, but for the precipitate reaction, it requires the optimal ratio antigen-antibody which occur in the optimum. Based on the sensitivity, the agglutination, it is more sensitive because there is a visible clumping, but in the precipitation reaction, it is less sensitive because the smaller complex may be harder to detect without enhancement. That's why this precipitation reaction, it is less sensitive. For example, about the agglutination reaction, that is blood typing, but for the precipitation reaction, examples like the radioimmunodiffusion. About the diagnostic application, to detect the large molecule or the particle often used for the cell surface antigen. But for the precipitation reaction, it's looking for the quantified soluble protein or the antibody which is mainly represented in the serum of the patients. That is the difference between the agglutination and the precipitation reaction. Let's talk about the question number 4, saying how is the radioimmunodiffusion test is performed. First of all, what is the radioimmunodiffusion? The radioimmunodiffusion, it is also called the single radioimmunodiffusion. Why is it called the single radioimmunodiffusion? It's because the only one of the antigen or the antibody is able free to move. The radioimmunodiffusion is the molecular immunological technique used to measure the concentration of the specific antigen in the sample based on the precipitation reaction between the antigen and the specific antibody. That means that this is the radioimmunodiffusion. It can help us to detect the quantity of the antigen which present in the patient sample based on the precipitation reaction between the antigen and the specific antibody. This radioimmunodiffusion is commonly used to quantify the serum protein, for example, like immunoglobulin, the complement protein, or it can also be used in the laboratory diagnostic. It is used to quantitate the antigen, to know the quantity of the antigen which is present in the patient's sample. Let's talk about what is the principle behind the radioimmunodiffusion test. As we discussed it before in this previous slide, we have seen that there is antigen-antibody interaction. The antigen, it diffuses the radially from the well into the agarose gel that contains the uniform concentration of the antibody. As the antigen moves through the gel, it binds to the antibody and forming the precipitate ring. The diameter of this proportion of the concentration of the antigen in the sample. It can allow us to quantify the antigen which is present in the patient's sample. Here also it is based on the antigen antibody interaction, but here the one of the antibody antigen, it can move in order to interact with each others. During performing this, the radioimmunodiffusion, it can take in the different steps. We are going to discuss one by one. The first step is the preparation of the agarose gel with the antibody. In order to perform this, the radioimmunodiffusion, we have the three kind of the solution. We have the solution containing antigen, which have the unknown concentration. The second, we have the solution containing the antibody, and the concentration of that antibody in solution is already known. And the third one, we have the solution containing the antigen, and concentration of that antigen, it is already known. Then taking the solution containing the antibody molecule is the added to the molten agar. We take that solution containing the antibody and pour into the molten agar. Then this mixture is then poured onto the glass slide and allowed to settle. You take that mixture of the antibody and the molten and you pour on the glass slide, and then after to be settled, we make the 4 or the 8 wear using the punch tool in order to make that wear on the agar slide. That the agar slide, it contains the constant amount of the antibody which is going to react with the antigen from the wear. That is the first step in the radioimmunodiffusion, where we prepare the agar slide which contain the antibodies. Then the second step is the loading the antigen sample. After making the well, we have to put in the sample in that well. Here there is the image which can showing the clearly how we are going to load it or to put that sample in the well. Firstly, here we have the first 3 way. We have to put in the sample which containing the antigen, and the concentration is already known. And in this, the last way, that is where we put the solution containing the antigen, but their concentration it is not known. That is what is written here. The known concentration of the antigen and unknown test sample are pipetted into separate way. In the first 3 where the standard antigen solution is added, and in the fourth where the antigen sample of unknown concentration is then added, and then that agar slide which containing the sample, it can make it to be incubated at the room temperature at a specific temperature to allow the diffusion, which is the 4th stage. Then after incubating, we incubate them between 24 hours up to 72 hours, and the antigen, it is started to diffuse out of their well in all directions. As the antigen diffuse outward from the each well, it encounter the antibody in the gel or the agar slide. Because the antibody molecule is uniform, when the antigen reaches to the optimal concentration of the antigen-antibody ratio, the visible ring precipitation form around each well. That means that in this third step, that is the where the antigen, it can diffuse out in the well and can bind with the antibody which is found on the agar slide. When the optimal antigen-antibody interaction to occur, the ring precipitation it is mainly formed. Then the next 4th step, it is measuring the diameter of that precipitate ring. After measuring that the ring formed, we have the diameter of the ring formed. It is correlated with the concentration of the antigen in the well. When you say it is the large ring, that means that in the well there is the large amount of the antigen. When we see the precipitate ring is the small, that means that in the well there is the little amount of the antigen. Then the last one is to quantify. In order to knowing the antigen present in an unknown sample of the patient, we have to comparing their ring diameter to the standard one. We have to take that the ring formed on the well which containing the solution containing sample of unknown solution and comparing it with the standard curve. Then it helps us to determine the quantity of the antigen which is mainly present in the patient. That is the diagram which can showing how it is done. And here, when the antigen radiate out or they move out, it can form the different link based on the antigen present in the well. That's discussing about the fifth question, which saying what is the principle of the ELISA technique. First of all, we have to know what is the ELISA. In the full word, when you say the ELISA, that is the enzyme linked immunosorbent assay. The ELISA is immunological assay or the biochemical technique which is mainly used to detect and quantify the specific antigen or the antibody in the sample. This is the technique. It is also based on the antigen-antibody interaction. It is widely used in the diagnostic, in the biomedical research and the quality control because it is the sensitive and specific and relatively simple to be performed. In the enzyme-linked immunosorbent, we use the enzyme conjugated with the antibody which react with the colorless substrate, and when the enzyme react with the colorless substrate, it generates the color reaction product, and that substrate which react with the enzyme, it is what we call chromogenic, the substrate. Let's talk about what is the principle behind the ELISA technique. The ELISA technique is based on the specific binding between the antigen and the enzyme conjugated antibody. And here, when that enzyme conjugated antibody bind to the antigen-antibody interaction, it can give the signal, and usually that signal which is formed is the color change, which can be measured quantitatively. When supposing that in the patient, we have the patient sample, and when that patient sample it is poured on the well, then adding the antibody after, then adding the enzyme linked the antibody, that the enzyme linked the antibody, when the substrate is added, the enzyme and the substrate can combine and developing the color, and that color it can be measured and giving the result quantitatively. Let me see the difference step which is involved in the ELISA procedure. The first, it is coating. When, for example, in the sandwich, we need the well which is coated with the antibody. First, we have to coat microplate with the either antibody or the antigen. Then the second step is breaking, that the microtite which is not coated, it needs to break with the unrelated protein to prevent non-specific binding. Then the third one is the incubation with the primary antibody which is not linked with the enzyme. Then after we have to adding the enzyme linked second antibody. The main importance of this antibody is to go to detect antigen-antibody interaction, and then enzyme become activated. And after enzyme becoming activated, that is the substrate is going to be added. That the enzyme it reacts with the substrate and to develop the color change, and that intensity of the color it is going to be measured, and depending on the intensity of the color, it can help us to correlate to the concentration of the target molecule which is present in the patient sample. We have the main four types of the ELISA. We have the direct, indirect, sandwich, and the competitive. The first direct, for example, in the patients come to the hospital and taking the patient sample. That sample pour it on the microtite well which is do not coated. Then after putting, we have to add the antibody which is linked with the enzyme. When there is antigen in the patient sample, it it forms the antigen antibody complex and the enzyme becoming activated. When the substrate it is added and the enzyme react with the substrate, it can developing the color. When that color measured, it can give the result quantitatively and can help us to predict whether the patients have the too much antigen in their body or they have no antigen. That is the direct ELISA. And indirect ELISA, it's look like the direct ELISA, but for indirect ELISA, we use two antibodies. The first, when the serum of the patient is pressed on microplate, then adding the antibody which is not coated with the enzyme, and then form when the presence of the antigen in the patient can form the antigen antibody complex. Then after washing, we have to add the second antibody which is linked with the enzyme. When there is antigen antibody complex, that enzyme linked to antibody become activated. Then after washing, adding the substrate and the enzyme react with the substrate to develop the color. When that color measured, it can give the result and it helps us to correlate the quantity of the antigen present. That substrate combined with the enzyme, it is what we call the chromogen. That is the second type, and the third one, that is the sandwich. In this third type of the ELISA, the first microtite is coated with the antibody. Then when we put the patient sample, when there is the presence of the antigen in the patient sample, it can react with the antibody and forming the antigen antibody complex. When that complex formed, they go and wash and to remove the excess serum, then adding the enzyme linked antibody. That enzyme linked antibody, it can go and binding to that complex and the enzyme becoming activated. When the substrate is added, it reacts with the enzyme and develop the color change. That color change also it can be measured and give the quantitative result. Let me sing about the competitive ELISA. Here, instead of the being microtite coated with the antibody, it is coated with the antigen. Before performing it, we take the empty container and then put the antibody and the patient sample together in that container and then incubate at the optimal temperature and the time. After incubation, we have to take that mixture of the patient sample and the antibody and pour onto the microtite well. Then after pouring on the microtite well, there is the remaining antibody which do not bind to the antigen, and then that antibody it can go and binding to the antigen which is coated on the microtite well. And when the washing occur, the antibody antigen which do not bound to the microtite well on the bottom, it's washed out and remaining that antigen antibody which is bounding to the bottom of the microtite well. And when the enzyme linked antibody added, it can bind to that antigen antibody complex which is bound to the bottom of the well. And then when the substrate is added, because already the enzyme is activated, it can react with the substrate and can developing the color. That is the different type of the ELISA. There is direct, indirect, sandwich, and the competitive. Here there is the image which can explain clearly how each different type it is going to be performed. Thank you so much for watching my video, watching my lecture. I would like to continue visiting my YouTube channel and following my discussion together in order to learn more about the immunology. Thank you so much for your kind attention. Thank you.